Everyone, and welcome to the ISCB's Youth Bioinformatics Symposium this year. My name is Olutoba Ojo, and I'm a current senior at Newark Charter School. And like many of you, I'm really passionate about bioinformatics. And being in an environment with peers this passionate about it is something you really can't get in the classroom. And I'm really happy that you all made it today. I'm very excited to be leading this first workshop on asking scientific questions. And we can just get started. So I'll first start with a brief introduction about what this workshop will discuss. Bioinformatics lies at the intersection of computer science and biology, but research in any field is about a lot more than just technical knowledge. Scientific research is about asking scientific questions and investigating them through hypotheses, experiments, and/or engineering. In this workshop, we'll get the opportunity to learn the scientific process and to learn how to come up with scientific questions that are meaningful, measurable, and practical. So I'd like to start with two quotes to really set the atmosphere for our discussion today. The first one is a quote by Thomas Berger, a notable American novelist, in which he states that the art and science of asking questions is the source of all knowledge. I really think that this quote proposes the idea that without questions, we can't further our understanding of subjects. And I believe that it can speak to the inquiry aspect of science, in which we're faced with a primary question, and then we constantly prioritize evidence and evaluate explanations for effective communication and justified reasoning. My second quote is by a notable French philosopher, Claude Lévi-Strauss, and he suggests that the scientist is not a person who gives the right answers. He is the one who asks the right questions. Asking the right questions is the essence of good science. Insightful questions can challenge accepted models and turn the way that we think about a concept. And of course, we'll always need a curious, inquiring mind to come up with the right answers. But the most exciting scientific discoveries would have never happened without the initial spark of someone asking a really good question. So I'll next give a summary of the scientific method. Now, I'm sure a lot of us are already familiar with these steps. It's a bit of a blast from the past or present in the way, considering the amount of times it's been drilled into our heads throughout high school. And honestly, over time, I had been a bit irritated by the amount of times I'd seen it restated in the labs. Yet reflecting, it's clear that the method had really been crucial to my understanding of research and my developing of scientific skills today. As without it, I probably wouldn't have been able to effectively reason and get behind the why of the research activities I've engaged in. For me, learning the scientific method over the years of schooling really outlined a rough idea of how science is done. The importance of the scientific method is that it lays out a useful framework for arriving at truth, and is characterized by using hard data, objective facts, and observations. It's also important to note that scientific method isn't necessarily a set of strict guidelines, as there are many models, and some models and research projects interpret the steps a little bit different and loosely. However, the steps mentioned here are arguably the most common and most accepted aspects that will eventually encompass all the specifics and details that a research project may have. The first step of the scientific method is proposing a question and supplementing it with background research. This step is foundational. So why is the question so important? This is due to the fact that the question will drive the rest of the scientific method and the research. To have a hypothesis, to have an experiment, to have data results, conclusions, and the like, a question must be made to start. Moreover, we can reflect from the previous examples and the two quotes I mentioned in the beginning and deduce that the practice of questioning truly is the root of science. So what drives the day-to-day work of scientists? Investigating, understanding, and developing explanations for how the Earth, the universe, systems, and etc. work. Recipes are a formula or procedure for doing or attaining something. Moreover, a recipe for investigation is built from a scientific question. So how do you come up with a scientific question? To come up with a scientific question, we should first understand the common elements that define what science really is. So what is science? I know upon seeing this, you may be thinking, oh, come on, we know what this is by now, but it's important to define it as it's crucial to the understanding of what questions can derive from it. Science has a goal of explaining the natural world. Moreover, a more textbook definition proposes that science is the experimental, hypothesis-driven investigation of phenomenon to elucidate patterns in nature, the processes governing the formation, maintenance, and changing of those patterns. So what makes scientific questions? An example of a good scientific question is one that is specific and can be tested using the scientific method. Another perspective is that scientific questions can be tested and evaluated using different steps, considering the fluidity of the scientific method. Another interpretation is that questions can be answered by science. However, scientists also ask questions when they're not necessarily expecting answers. Such questions are often advice or a suggestion of an alternative hypothesis or a problem raised. In other words, questions and answers serve to deepen the understanding and encourage the exchange of ideas. We should also be careful of using questions that may be subject to different opinions. For example, if you were to ask, who is the best scientific researcher? Like, I know you can say me, and I'd be flattered, but because we may receive varying answers from different people, the questions are considered subject to opinion and cannot necessarily be answered by science. So what can scientific questions do? Scientific questions can suggest a new direction for research. Maybe there is a different way that a person thought about going about it. They can also challenge research interpretations or suggest other possibilities. They can ask about previously established facts and information in order to inquire about any connection with other research, and they can draw comparisons between research. Now, for proposing research and scientific questions, how can we ensure that scientific questions are meaningful, measurable, and practical? First, we should describe the important unsolved problem. Next, we should emphasize the challenge and the idea of the unsolvedness. We should also describe other approaches to the problem besides ones that we've made. It's important to note that we should also provide deep analysis of the overall problem and challenges, and that it's finding and forming scientific questions aren't necessarily straightforward, and it's actually very common to struggle with them. But an early practice into making scientific questions helps us mature in many ways as potential researchers, and learning how to ask a question is an incredibly good skill. Lastly, we should describe the problems that are still unsolved and what potential there is to solve them, typically via our research. Now, for the foundations to making scientific questions in bioinformatics. Bioinformatics is a very broad and diverse field, and it has several subcomponents. Examples of those include structural biology, genomics, cheminformatics, phylogeny, and etc. So we should really do a deep dive and get acquaintance with the different subcomponents that we may come across. I'd like to note that having a biology background especially can help us fall in love with these topics and give us more grounding on what we can make of them. We should also read recent research articles and see the trends of research, because thereafter we can really develop curiosity and interest and see which topics match our skill sets. Moreover, we should take the knowledge that we already have and develop questions about what else we may want to know. I'd also like to re-stress reading research articles, because you can really develop an appreciation for the current problems and be up to date with the things that are surrounding the world of bioinformatics today. For my own research that I did at a program in Pitt, I was asked to read a lot of articles just to gain a strong understanding. There are several high impact journals and other resources that you can look at to find some research articles. A point not mentioned here, but that is also good, is consulting professors. It's always good to look up to people that have gone through the bioinformatics journey and that are actively doing research in them. They're experts in the field, and they may be able to provide valuable insights regarding research projects. Now, for forming bioinformatics questions. Bioinformatics is often thought of as the applications of computer science principles to biological problems. Therefore, in forming questions, we should always have a clear definition of our biological problem. Subsequently, we will discover new methods that will be used to solve our problems. Now I'm going to transition into research and forming questions in research and papers. For the next couple of slides, I'll be stressing the importance of reading research. We'll be discussing expanding our knowledge. Well, the purpose of reading research is to expand our knowledge so that we're better informed of how to solve a problem. Having good habits for reading scientific literature as well can set you up for success because you'll be able to identify new research questions and fill in the gaps for things you may not understand initially. So the earlier that we have this exposure, the better in the long run. When we look at research papers, we should strive to formulate questions from them. For example, it's important to possibly ask the following. What is the paper about? What is said in detail? What type of article is it? Journals publish different types of articles, including methods, review, commentary, resources, and research articles, as well as other types. There are often different formatting requirements and expectations for understanding the content. We should also ask, what do they do? And in doing so, we'll be discovering their approach and their methods to their research. A follow-up question to that would be inquiring why it was done in that way and its context within the field. And then we should finally ask, what can we make of it really? After reading research, we should always have a takeaway. What are some action points that I can do after reading it? Okay, and here I provide an image of the basic structure of a research article, starting with the abstract here, that essentially summarizes the whole study with a short introduction in the first 2 or 3 sentences, talking about the different attempts made in the study, and then it jumps to the methods, which builds upon the introduction. The introduction talks about the background of the study and things that we should know beforehand. The methods show the different components that were brought in together for the experiment, and thereafter were brought to the results, which summarizes just the effects of the experiment and what we can take away. And then it closes with a discussion, of course, and a conclusion, and finally stating references. I'll next speak about reading research articles. I'd like to put a forewarning that research papers are often sometimes difficult to understand, especially when you're new to the field, and it can really require a learning curve, but constant practice really enables further understanding. To truly understand a scientific work, you will often need to look up a term or dig into supplemental materials or read one or more cited references. Likewise to how our scientific reading skill develops, this process also takes time. For my own research in a Pittsburgh program, I had to read an article over and over 3 times. The first time I just read without the pressure of understanding or critiquing the work. And the second time, I tried to understand gradually. And for the third time, I took notes so I could understand it comprehensively. The simplest way of reading a research article is to start by reading the abstract first. Reading the abstract carefully and understanding what the study is about, how it was carried out, and what results and conclusions are mentioned in the lines of the abstract are foundational to our understanding. After getting a complete idea of the study, you can go to the conclusion part. You can read the conclusion and understand them, and if you find the study interesting, you could read the results section, followed by the methods and materials, and then go to discussion. Furthermore, if you would like to know a lot more about the topic of the study, I would encourage you to read the full introduction part. And if you want to delve really, really deep into it, I would read it all in its entirety from the top. So now I'm going to transition to a more hands-on. In this link here, I provided an article, research paper. So I'm just going to set a timer for 3-ish minutes in which you guys can just skim through the following research paper and then try to determine a question that the research aims at answering. And just remember what makes scientific questions, as I mentioned before. And you could possibly propose your own question. Then after that, we can paste our answers in the chat and collaborate. So I'm just going to set a timer for, I guess, 3 minutes. The link is here. If you guys run into anything, just let me know. And feel free to ask questions as I'm going along. I know I'm kind of pushing through here. Let me mute. Okay, I think we can rejoin here. All right. Does anyone want to share their questions that they came up with? Uh, I don't think I can simultaneously see the chat, so let me see if there's anyone. Okay, we could still move forward. Let me reshare. One question that, oh, oh, one question that we could have come up with is, let me make sure I'm on the right slide here, actually. Apologies for that. One question that we could have come up with from reading the article is we could ask how effective are different next generation sequencing methods on molecular epidemiology practice? It's pretty broad right now, and I know I kind of gave like limited time. Ordinarily, you would have more time to fully analyze an article, but just skimming through the abstract and conclusions, we could probably pick up on some ideas of what to ask. We can probably we can do the next exercise as well, but let me just skip to my textual analysis for it. Uh, let me open this actually. So it's this article titled Abstract Learning via Demodulation in a Deep Neural Network by Andrew J.R. Simpson. And after skimming through it, well, I read it comprehensively, but skimming through the abstract, we could be led to here. I just took some quotes from it. Since the brain is adept at abstraction, it is anticipated that deep neural architecture might somehow capture abstract representations. However, it is not presently known or how, or even if abstract learning occurs in the DNN. So I just thought that this comprehensively covers really the purpose of the article and what he's aiming at discussing, which brought me to my research question that I was able to derive from it. How does abstract learning take place within a deep neural network, or DNN? And so this study demonstrates that the DNN learns abstract representations of data via a process of demodulation. With a highly biased sigmoid activation, the researchers were able to discuss the question of abstract learning in a quantitative way and discover if demodulation is a factor in DNN learning. In conclusion, these results have broad implications for how deep neural networks are interpreted, designed, and understood. Furthermore, our findings may provide insight into the exceptional abstract learning of the human brain. I again know I'm throwing a lot at you right now, but the takeaway from it is essentially just taking pieces from the abstract and different parts of the article to really deduce what the author is trying to say and what research questions that we can form from them. I'm next going to go over a live example from my summer research at the University of Pittsburgh's Medical Center for the Hillman Cancer Center Academy program. So my project was single-cell RNA-seq cell type annotation. I was mentored under Dr. Uma Kondran and Alexander Chang. And for some background, when we go into the complexity of an organism, our goal is to really discover what makes up the organism, examining the insides of a cell, and observing how many RNAs it's able to make. Single-cell RNA sequencing is a newer method to analyze RNAs in individual cells, leading to a more accurate inference of identities for the cells in a sample. It requires complex computational methods in order to infer as accurately as possible. It's becoming more popular as it allows scientists to ask more questions and propose more answers for next generation sequencing. The differences in gene expression correlate to different proteins and in turn different cells. So here we have our single cell RNA-seq pipeline, which is essentially just the usual workflow for analysis studies. First, we have quality control, then alignment and quantification, low quality cell filtration, normalization, HVC selection, dimension reduction, clustering, and finally, cell type annotation, which my project revolves around. So what is cell type annotation? Cell type annotation is the usage of computational and bioinformatics methods to assign identity to cells. It can be broadly categorized into these types here, which is marker gene database based, correlation based, and supervised classification based. The marker gene database uses a gene list. The correlation base uses compares gene expression value from the data set to a reference data set. And the supervisation classification based method uses machine learning methods. So that will bring us to our methods section. Our primary question was, how do cell type annotation tools affect cell type annotation? For our study, we used single cell data from the Pangolo database, which is a single cell repository database across different mouse and human cell data. From it, we downloaded a mouse motor cortex data file with over 10,000 cells in it. And most of them, as we can see here, were unannotated and unknown, as the red correlates to the massive amounts of unknown. And for our annotation methods, we compared 3 different methods, each belonging to the different types of annotation as we saw back here: marker gene based, correlation based, and supervised classification based. The marker based was SC type, the correlation based was single R, and the classification based method was a Surrey classification, or azimuth. These were all web-based or R-based, which is a programming language. So going into results for azimuth. First, I'll discuss what all of this means. I know it's a lot. Azimuth is a tool built by the HubMap Consortium, and it has a normal reference data. It is based on Surrey's methods from the NYU Genome Center Satija Lab. We downloaded the mouse cortex data from Pangolo and uploaded it to Azimuth, and used the brain cortex data in Azimuth as a reference file to compare. The UMAP figure shows the Pangolo data mapped to Azimuth's brain reference cell types. In other words, just comparisons with one another. The next table is a contingency table, which shows how many cells from each of the unknown clusters also map to different cells in the azimuth references. And again, to reiterate, just comparisons as we can see here. For example, as a positive control, all the cells labeled in microglia in Pangolo are in fact labeled as microglia in azimuth also. You can think of them as percentages, as you see here, like 100%. And similarly, all the parasite cell in Pangolo are also parasites in azimuth as well, as we can see here. So what about the unknown or unannotated clusters in Pangolo? As you can see in this table, for cluster 11, which is unannotated in Pangolo, 90% of the cells are mapped as MEIS. In cluster 1, in cluster 1, which is unannotated in Pangolo, 83% are mapped as L6CT, as we can see the correlation there. So this contingency table gives us some idea of the number of cells from each of from each Pangolose cluster's mapping to the azimuth reference database using Surratt's methods for cell type identity assignment. So what did I just say? Again, it's just comparisons between the single database and the computational method. Shown on the bottom right here is a figure where the y-axis is the prediction score here and the mapping score. So the analysis can really become complicated. The higher the prediction score, the better, and each dot represents a pangolin mouse cortex cell. As you can see in microglia here, and the parasites in general have a good representation, and they have higher prediction scores, which means what's considered microglia is also considered microglia by azimuth as well, and it predicts it with a high confidence. However, when we look at MEIS, we see it's unlike the other ones too. The prediction score has a range of values, which means we can't tell for sure if the unknown clusters were labeled as MEIS are in fact accurate or not. Therefore, we have to do more digging into the data to understand the annotation more overall, but we can conclude that some of the cells in some of the clusters in Pangolo are annotated by azimuth, and mostly as MEIS2, LAMP5, and L6CT. We next tried single R on the same Pangolo dataset and using the same brain cortex reference. Although not shown in these figures, we determined that the cells in Pangolo are annotated as neurons at the tissue level, which is good, meaning the unannotated clusters are not all bad data. Now looking at the specific cell types, single R is a correlation-based method, and the color on the left-hand heat map here is an indication of the correlation. The yellow is the best correlation, and the blue is the lower correlation, as we can see with the key here. First, if we look at the small yellow box on the bottom left, it shows that Pangolo cells correlate very well with the astrocytes. And it's the same thing for parasites as well. Therefore, in concordance with azimuth, cells that are labeled as astrocytes and parasites by Pangolo are mostly likely also labeled the same in single R. Now let's look at this big yellow bar at the top here. This means that a lot of the clusters are labeled as MEIS2 and with a high confidence. We cannot really deduce that they're unannotated cells in Pangolo. The contingency table on the right here shows that almost all of the unannotated clusters on the ones that are labeled the NA are also annotated MEIS by single R. At the bottom, we can also see that some are also called astrocytes, but unlike azimuth, they're not L6CT or LAMP5. So in essence, single R and azimuth agree on the annotation of the known clusters in Pangolo, such as parasites, but not on the unannotated clusters. But again, we would have to look at the correlation score more carefully to understand the confidence in the prediction of these annotations. The last tool that we utilized was SC-type, which is marker-based. It produces a nice colorful output, or bubble plot, with the size of bubbles corresponding to the number of cells. However, the bubble plot, although visually nice, is hard to understand, and we have to dig deeper into the metrics and understand the outputs. The middle figure here, the contingency table, shows that many of the unannotated clusters are called astrocytes by SC-type, as we can see the correlation there. The chart on the right is what we call a normalized score. Shows what we call a normalized score, and it's one way to assess or think about the accuracy of the annotations. For example, clusters annotated as parasites or microglia in Pangolo are also annotated the same in SC-type and with high scores, as we can see here. Other clusters, the unannotated clusters, have lower scores, but still do pass the SC-type thresholds for annotation, or what is considered acceptable. Again, for the first two tools, we will have to dig deeper into the score to understand the accuracy of the annotations. So based on this, we can conclude that the usage of different single-cell RNA sequencing tools, annotation tools, can lead to different interpretations of results. And although all tools agree with Pangelo on the annotation of astrocytes, pericytes, and endothelial cells, they do not agree on annotation of the unannotated clusters. For example, Asmus labels some of the unknown cells as MEIS2, LAMP5, L6, CT. Single R labeled a majority as MEIS2, and the SC-type method labels them as astrocytes. So more work needs to be done in order to assess the confidence of all three of these tools. Another point is that annotation is a challenging subject within single-cell bioinformatics. It's important to understand the variability amongst different tools and their algorithms. For further studies, it would be good to experiment with different examples and different reference files to explore the variability of these tools. So what did I say again? To summarize the research, starting from the background, and I'm making comparisons to how we analyze scientific literature, starting from the abstract, the methods, to conclusion, and I'll get to all of it. So all living systems are composed of cells that we know, and they all contain crucial RNA molecules with important information and processes. In identifying and quantifying the expression levels of RNAs within a cell by technologies such as RNA sequencing, scientists can infer the identity of a cell. Single-cell RNA sequencing, or scRNA-seq, is a newer method that enables scientists to quantify the RNAs in individual cells and therefore inform more accurate identities for the cells in the sample. It really enables scientists to deeper understand the complexity of cell types. And we use the computational methods, as I said, and we use those three different tools. Reviewing again, the methods that we used were, we first initially retrieved the mouse motor cortex data from the Pangelo database, and then we annotated them using azimuth, single R, and SC type. In contrast to the azimuth method, single R and SC type require the normalization of data as a step prior to running completely. And again, all tools used the same reference mouse motor cortex data and was retrieved from the Pangelo database. As we had seen, azimuth used an annotated mouse motor cortex reference as a basis for mapping and outputted two plots visualizing cell type prediction. Likewise, single R used reference transcriptomic datasets of pure cells for its annotation and outputted a heat map graph distinguishing the varying levels of expression among cell types, as well as a violin plot visualizing the distribution of cell type expression. SC type used a combination from its cell marker database and outputted a cluster graph with cell types and a mapping classification graph. And in conclusion again, we're able to conclude that the usage of different computational methods can directly lead to different interpretations of data. Wrapping it back, we should be able to draw possible questions from this research. For example, how can the usage of different single-cell annotation methods lead to different results is an example of a valid question. In addition, how does an individual mouse motor cortex dataset affect its cell type annotation is another question that allows us to think more on the topic. What measures can be made to increase the confidence of the cell type annotations is also another scientific question that allows us to further try to find ways to study it, the ways to increase the confidence for cell type annotations further. And with that, I'll be closing and just summarizing some key takeaways from this workshop. The art, as I said in the beginning quote, the art and science of asking questions is the source of all knowledge. Reading and contextualizing research enables us to expand our personal knowledge on the topic, so we are better informed in our experience on how to solve the problem. So I just wanted to give special thanks Special thanks especially to Darsh, as well as the International Society for Computational Biology, as well as the University of Pittsburgh's Hillman Cancer Center Academy. And those are my sources. If you guys have any questions, feel free to let me know. I haven't been checking the Q&A. I don't know if there's any, but thank you.
Great, thank you so much. I just reminded people to post in the Q&A as well, so we're just going to take a quick there, see if we have anything new come up.
I saw a couple questions pop up while Toba was giving his presentation. So,
perfect. Thanks, Darsh.
Let me take a look. All right, here's the question from Nasra. I think this popped up when you were talking about reading scientific papers. She was asking how to deal with constantly getting hung up on unknown terms / jargon when reading these papers. Sometimes it gets pretty confusing or overwhelming.
To that completely. In fact, the first time I was doing this, I was completely lost. But I think a lot of it just comes from searching it. Research builds on itself in the sense that you can find different articles that relate to one another. So again, I think really just searching for them and trying to build a gradual understanding will enable you to understand it further.
Awesome. I think there was one more. Let me see if I can find it. How does our research questions differ when it comes to engineering projects?
I'm kind of struggling to understand that question.
I mean, I can try to answer this too. I think she's asking like, how is it different when we are trying to solve scientific inquiry questions versus engineering design questions? And I mean, I feel like those two are a lot more intertwined than they seem. Engineering projects are often really trying to answer a scientific inquiry question. And so when you're asking a scientific inquiry question, you might ask, how does X affect Y? Versus when you're doing an engineering project in research, I feel like oftentimes how can I build a tool that allows me to further understand the role of X and Y? Yeah, so that's it. Toba, do you have any other ideas on how engineering questions differ from science inquiry questions?
I think you nailed it on its head right there.
All right. There was one more. This is a little more technical question, but I lost it. Okay, there it is. So someone's asking, can you talk more about batch normalization, as this process would be quite important for this?
I'm not familiar with batch normalization, but what I'm seeing now, it seems to be a artificial intelligence and machine learning method or process, I believe. Is that correct?
I think honestly, I'm not too familiar with this process either. So, I the Q&A chat right now.
Sorry to interrupt. Did you guys want me to interject a bit on batch normalization?
Go for it.
Sure.
All right. So for those of you who aren't aware, my name is Seth Mulholland. I work for ISCB. I have my PhD in bioinformatics before I with the society. So normalization is a very important process. When you get your data, before you can really compare apples to apples, you need to normalize. You need to get it to be set to a same standard. An easy example, if something is measured in feet and another thing is measured in meters, normalizing would be converting the feet to meters or the meters to feet, just so that you're looking at them side to side. Now, this is hugely important on your data, because if your data is disparate forms like that, you'll get weirdness. You can get novel output that isn't actually novel. It's just a product of the differentiation between your data types. Now, as you said, batch normalization is used in like neural networks, in AI neural networks, in machine learning, things like that. And it is just as important, it's actually really more important, because it's used in the training of these networks to help them move forward and pick up on any kind of patterns, any kind of algorithms they might be designing. You have to have a really good data set to build them off of. And so keeping all of your data in batches is just standard. If I collected on day one, that could be my batch. If I collected on day two, that's another batch, things like that. But you need your batches to normalize within themselves and within each other. And it is huge. It is convoluted, and especially when you're dealing with things like AI neural networks, the math behind it is massive. And it is, to be candid, it's annoying. If you make a mistake, it will manifest itself later, and a lot of your results can be questioned because of a mistake early on. The specific process for it, I could go on for hours, and really it is its own specific subset of computer science study of exactly how do and should we normalize these things. But the basic idea is you need your data all to be on the same ground before you start analyzing it for differences, to make sure that the differences you're seeing are meaningful to that data set and not just an artifact of the kinds of data you have.